I spent about 3 hours annotating this worksheet for you, this key. I added a lot of definitions. I hope they help. So we're just going to go ahead and go over it right now. Just like for every worksheet and every chapter in the book, you should memorize your vocabulary words. And then for biochemistry, there are certain structures that you should practice drawing over and over again until you feel comfortable with them. So here we're talking about sugars. So monosaccharides are just one sugar. You should know glucose and fructose. And then mannose is also one of the most common ones. Glucose is an aldose, so that just means that there's an aldehyde group at the end. Fructose is a ketose, and that means that it has a ketone instead of an aldehyde group. And then we have some disaccharides that you should also practice drawing: sucrose, maltose, and lactose. And then what each one is composed of: a glucose and a fructose, a glucose and a glucose, a glucose and a galactose. So you should know how to draw these guys. And then we have polysaccharides, and that's when you have like thousands of glucose monomers with a few other things. So some of the most common ones are starch and cellulose. All right, for question number 1, they want you to go from Fisher to Hawthorne. So definitely watch the video, but I'm just going to go over a few things here. For the Fisher projection, you always want to start numbering with your aldehyde group at the top. And then you number your carbons. Here's 1, 2, 3, 4, 5, 6. So this is D-glucose. So notice that for carbon 2, the alcohol group is on the right. For carbon 3, the alcohol group is on the left. For carbon 4, it's on the right. And for carbon 5, it's on the right. Okay? Now, you have to know when you're going to go from Fisher, linear, to Hawthorne, circular, something happens, okay? So here at this 5th carbon, that's like the most important carbon for this transition, because the alcohol group that is attached to it, the lone pairs on the oxygen, they attack this first carbon. And then a few steps happen, and then you end up with this circular structure, okay? So you'll learn about that when you watch Shkala's video. Let's see. Okay, so then there's a few other rules. Anything on this right-hand side is going to be facing downward on the Hawthorne, and anything on this left-hand side is going to be facing upward in the Hawthorne, except for this stuff here at the bottom, because a rotation occurs and then this changes. Okay, and now D. What are they talking about with D here? So the D refers to the last chiral carbon at the bottom, the one that is farthest away from the aldehyde, okay? So when I first saw this, I was like, oh, it's got to be this last one. But if you pay attention and you know your definitions, then you know that chiral carbon, this is not a chiral carbon. A chiral carbon has all 4 sides different. So it is attached to 4 different groups. This carbon right here has 2 hydrogens. So it has like a pair of twins. And that means that it is not a chiral carbon. So that's carbon number 6. We move up to carbon number 5. And we see that it has a hydrogen attached. It has a hydroxyl. It's got this CH2OH. And then it has this giant group at the top. So that's how we know that that is a chiral carbon. It has 4 different groups attached. So that's the one that we're going to look at to determine whether it's D or whether it's L. So once we find our last chiral carbon that is farthest away from this aldehyde group, we look at the alcohol group that is attached to it. And then we say, okay, if the group is on the right, then this is a D. If the group is on the left, then it's an L, okay? Now, once you have this glucose molecule memorized, now you can look for epimers of glucose. So what is an epimer? I wrote it for you up here. Epimers are diastereomers that differ at only one chiral center. So an epimer of glucose has several epimers, okay? Now, it just means that the structure is exactly the same, but it's only different at one of the chiral carbons. So you can see that these four carbons are in purple. Those are all chiral carbons. And if we look at carbon number 2, we notice that the alcohol group was supposed to be on the right, but instead it's on the left. And that's the only thing that is different from this structure to this structure. So this is D-mannose. So we know that mannose is a carbon 2 epimer of glucose, okay? D-mannose is a carbon 2 epimer of glucose. That's how you do that, okay? And then it's still D because this carbon down here is still the last chiral carbon that is farthest away from this aldehyde group. And that alcohol is still on the right-hand side, which means that it's still D, okay? Now, to go from linear to the cyclic, you first add a mild acid to activate the aldehyde group. So that's how you go from linear to circular. And then only a little bit of glucose molecules exist in their linear form. Most of them actually exist in their cyclical form because it's energetically favorable, okay? So only about 1%. Well, they go from 1%. I think in lecture they said less than 10%, but it's only a small amount. Okay, so once we are in the cyclical form, guess what? To determine D or L, it's different. Okay, and we have to absolutely know these two. So over here we cover that to determine whether it's D or L, you have to look at the last chiral carbon away from the aldehyde group. And if that alcohol group is on the right, it's D, and if it's on the left, it's L. Well, when you're in the circular form, all you have to look at is your 6th carbon, okay? So if this group is up, then it's D. If that group is facing downwards, then you have L, okay? Now, okay, so now this alpha. How do you know if something is alpha or beta? Well, we look at the anomeric carbon. This is our anomeric carbon right here. If the alcohol group can either face down or it can face upwards on the anomeric carbon. So notice that I'm being very repetitive for you here, and that's because at the end of this discussion, I'm hoping that you'll have like half of your vocabulary words memorized. That's why I'm being repetitive on purpose, okay? So, okay. This anomeric carbon has the alcohol group. It can face down or it can face up. So to determine whether something is alpha or beta, you look at your carbon number 6 and you look at your anomeric carbon, the group that is attached. If they're both going in the same direction, then that is cis and it's going to be beta. If they're going in opposite directions, then it's going to be trans and it's going to be alpha, okay? So in this case, we have our 6th carbon that's going up. And we have the alcohol group that is attached to this anomeric carbon that is going down. So they are going in opposite directions, so that means that it's alpha. Easy, okay? What do we have over here? Okay, that's it for 1. And now we have B. Draw the Hayworth projection. Draw the Hayworth projection of the beta furanose form of Fischer projection. Okay, so here we have a ketose. Look at this. carbonyl is in the middle, and then this is an R group, and this is an R group. Okay, so when it folds into its cyclic structure, you notice that it's a little bit different. When you watch Shkola's video, you're going to understand why. Okay, but for now, let's do this. Again, all you do is number your carbons. This is still carbon number 1, carbon number 2, 3, 4, 5, 6. Okay, everything on the right-hand side will be placed downwards in the cyclic form, and everything on the left-hand side will be placed on top in the cyclic form. Okay, so you can always start your problems like this. You draw your structure in a straight line, and then depending if it was on the right-hand side, you draw that group on top or down below. If it was on the left, you draw it on top. Okay. Okay, and then something else that we have to notice, I'll show you down here. Our carbon number 1 moved. Look at this carbon number 1. It's now in this location. So be very careful when drawing these structures when you have a ketone, okay, instead of an aldehyde. So your carbon number 1 is over here. Your carbon number 2 is now in this position. 3, 4, 5, and 6. So all the other carbons are in the same position, but carbon number 1 and 2 are a little bit different. So just practice drawing this over and over again, and you'll be fine. And then we also have to notice that this carbon right here, now this guy is our anomeric carbon. So it's still that position that is our anomeric carbon. And what we care about is the alcohol group, whether it is up or down, okay? So in this case, that alcohol group is up. And then we look at our 6th carbon, and that one is up. They're both facing the same direction, which means that this is cis and it's beta. Now, we have to determine if it's alpha, or, if it's A, L, or D, the configuration. So we did the whole alpha beta, we determined it was beta. Now we want to know if it's L or D. So to do that, all we do is look at the 6th carbon. If it is up, it is D. If it is down, it is L. So we see that it is up, so it is D. Okay? Easy. The more, we're going to keep practicing this, so it'll get easier and easier for you. Okay. So here I added some more vocabulary. Let's see. Okay, so what is a reducing sugar? A reducing sugar is a sugar that can be oxidized. This includes any sugar that contains a hemiacetyl group. Okay, an example is lactose. So lactose is a reducing sugar because it has a hemiacetyl group. So a lot of students don't really know what a hemiacetyl group is. So basically, this is all you look for in your structure. If you have your carbon here and you go up, you should have a hydroxyl. It can go up or down, but you should have a hydroxyl. And then when you go this way, you should have an O and an R. This is a hemiacetyl group that can be, it can open up to form a free aldehyde. Okay, so a non-reducing sugar is a sugar that cannot be oxidized. So if you don't see this group, then it can't be, you have a non-reducing sugar that can't be oxidized. An example of that is sucrose. It doesn't have a free hemiacetyl group. So let's see here. You're going to be drawing, let me erase this so that I can redo it, and then it'll be easier for you to understand what's happening. Okay, so they want you to draw these sugars. So in your book, they show you what glucose looks like and mannose looks like. You know that glucose is the first sugar and mannose is the second. So the first thing you should always do is number your carbons. So we already know that this for glucose is number 1, this is number 2, this is number 3, this is number 4, This is number 5, and this is number 6. So it's important to note that in this ring structure, there's 1 oxygen and there are 5 carbons inside of the ring structure. This 6th carbon is not inside of the ring structure, it is outside. It's very important for you to notice that because it's going to help us understand some definitions down lower in the on the discussion worksheet. Okay, so 1, 2, 3, 4, 5, 6. All right, and then let's number these guys over here. We have 1, 2, 3, 4, 5, and 6. Okay, so we see that this connection is happening between carbon 1 and carbon 6. Okay, now let's determine if this connection is alpha or beta. Okay, so let's look at carbon. The important carbons to look at to determine if something is alpha or beta is carbon number 6 and carbon, this anomeric carbon. Okay, so this O used to be the OH. Okay, so that's the one we care about. And we see that it is facing downwards. Okay, and we look at carbon number 6, that one is facing upwards. Okay, so these guys are opposite. They're trans, so this is alpha. Okay, and that's how you do that. Now, is this D or is it L? Well, for that, we just look at carbon number 6. If it's facing upwards, then it is D. If it were facing downwards, then it would have been L. Okay, so that's how you can figure out how to draw this when they only give you words. You just have to know the definitions and a few simple rules. Okay, so for mannose, they're also saying that it's D. So we look at carbon 6 for mannose, and if it's up, then it's D. If it's down, then it's L. Yeah, L. Okay. Oh yeah. So then we look to see if there's a hemiacetal group. Okay. So this right here is the anomeric carbon. Okay. And then if up here we see that we have an OH group, and then over here we have the O, R. So anything attached to this O is that R group. So we have an OH and an OR. That is our hemiacetal group. And that's how we know that this is a reducing sugar. It can be oxidized. Okay. Okay, let me check the chat to see if there's any questions. Okay, looks like everyone is good for now. We'll keep going. Draw the structure of gentobiose. So it's D-glucose, beta-1,6, D-glucose, okay? So I'm going to do the same thing. I'm going to erase this. Okay, I'll just leave the numbers there. Okay. Okay, so they want glucose and glucose. So you're just drawing 2 glucose. And they want the first one to be D. So to determine if it's D, you look at carbon number 6. So we draw our 2 glucose, we number our carbons, and then we go look at carbon number 6. They want this to be D, so that means that this has to be up. Okay, now carbon number, the second glucose is also D, so that means that carbon number 6 over here should also be up. Okay, now the link, this glycosidic bond between the first glucose and the second glucose should be a beta between carbon 1 and carbon 6. So This is carbon 1 of the first glucose, and this is carbon 6 of the second glucose. So that's the bond. That's how you know which carbon to which carbon the bond should go to. Now, it has to be beta. So what does it mean when something is beta? It means that they should be going in the same direction as carbon number 6. So our carbon number 6 was going up. And if this is going to be beta, then this link, this alcohol group that was right here, should also be going up. And so you can see that it goes up. Okay. So that's how you draw that. And that's a beta bond. And that's it. That's all. So you can see that once you get to know your rules and your definitions, it's easy. So here we have 3 different disaccharides. So we have lactose, sucrose, and trehalose, okay? So let's just check them out really quick individually. So here, the 6th carbon, we see that it goes up, and we see that the O, or the alcohol group for the anomeric carbon, it's also going up. They drew it a little bit differently here than they did previously, but you can still see that when you first start, it's going up, okay? Because it could have gone down this way and then like that, but you can see it's going up. So both of these going up means that it's cis and this is beta, okay? Now, is this D or L? Well, we just look at carbon number 6, it's going up, so it's D, okay? And over here, this is also up, up, so that's beta, and this guy's going up, so it's D, okay? And what else can we look for? Oh, okay, this glycosidic bond is between carbon 1 and carbon 4, okay? Now, do we have the hemiacetal group? Well, we have our OH, and then we have the OR. Yeah, we have a hemiacetal group, so this is a reducing sugar, okay? Now let's look at sucrose. Well, we go up, so it's D. And then here we go up, and then on the anomeric carbon, it's going down. So these guys are opposite. That means it's trans, and that means it's alpha, okay? Here we have, what do we have? Okay, so look at the numbers. Normally our carbon number 6 is up here, but in this case, they flip the sugar around, and so this is carbon number 1. 1, 2, 3, 4, 5, and 6. So this is sucrose. And the sugar is flipped around. And so we still have to look at carbon number 6. Okay, so carbon number 6 is down. And then this is the anomeric carbon here. And it is going down. Okay, so that means that they're both down. So this is cis and this is beta. Okay, now we have to determine if this is D or L. Well, carbon number 6 is going down, so it's L. Okay, so this is a really good example of how you can be tricked on an exam, okay? So sucrose is a really good example. So as long as you remember your rules and your definition, you know that you have to look at carbon number 6 and the alcohol group on the anomeric carbon. The rules are the same. If they're both going down, then they're going in the same direction, and that means it's cis, and that means it's beta, okay? All right. Oh yeah, let's see, do we have, okay, trehalose. It's 2 glucose molecules. Carbon number 6 is going up, so that means it's D, and then for the anomeric carbon, it's going down, so that means these guys are opposite, so that's trans and that's alpha, okay? So over here we have carbon number 6 is going down. Okay, so this is another good example. Now you see, we've always seen the carbon number 6 go up, but now carbon number 6 is going down. And then the anomeric hydroxyl group right here is going up. So this is trans and it's alpha. Okay, so now let's look at carbon number 6. It's going down, so that means it's L. Okay. But really, when they ask about that stuff, they usually just focus on this, the first one. Okay, so now do we have a hemiacetyl group? Well, we have our OH here, right? But then when you go to the left, there's supposed to be an O here, and then an R. So we do not have a hemiacetyl group because we don't have that O there, okay? So that means that this is not a reducing sugar, okay? So now we're going to go ahead and answer some questions. A pentose, do we have a 5-carbon sugar? We do not. So when I first saw this, I was like, oh, this guy's got to be a 5-carbon sugar because the ring is smaller. Well, let's check it out. We have 1 carbon over here. Oh, yeah, 2 carbons over here, 3, 4, 5, and carbon number 6 is down here. So there's 6 carbons. The ring structure is smaller. There's only 1, 2, 3, 4 carbons inside the ring, but they're not asking you how many carbons are inside the ring. They're asking you like a pentose sugar. That would mean that there's 5 carbons overall. But in this whole sugar molecule, there's 6 carbons overall. This is why it's really important to have your definitions really, really sharp. Okay? And over here is a lot more definitions. Well, they're asking if you have an aldose sugar. An aldose has an aldehyde group at the end. So all of ours had an aldehyde group at the end. Now they're asking you about a pyranose. A pyranose is a 6-membered ring, but it only has 5 carbons inside the ring. The other atom in the ring is an oxygen. So let's check that out. Okay, so this qualifies for that definition of pyranose, okay? So it's a 6, what did they say it was? It's a 6-membered ring, but only 5 carbons are inside the ring. Okay, so 6-membered ring, 1, 2, 3, 4, 5, 6. There's 6 members inside of this ring, but only 5 of them are carbons, and 1 of them is an oxygen. That's a pyranose, okay? Now, furanose. What's a furanose? There it is. A furanose is a 5-membered ring, but it only has 4 carbon atoms in the ring and 1 oxygen. So furanose is a 5-membered ring, but only 5 carbons in the water. So we found this guy right here, right there. 1, 2, 3, 4, 5. 5 members in the ring, but only 4 of them are carbons, and 1 of them is an oxygen. So this is a furanose. Okay, what about a ketose? A ketose has a ketone instead of an aldehyde. Okay. L stereoisomer. So we remember how to do L versus D, both in the linear structure and the cyclic structure. Let's see. Okay. Name the following monosaccharide by placing the appropriate terms alpha beta, LD, pyranose, or furanose. Okay, so for this first one, let me erase that so you guys can see better. Okay, so it's just more repetition and more practice of everything we've done. The first thing we do is we go ahead and number our carbons. Okay, so that's just 1, 2, 3, 4, 5, and 6. Okay, so the next thing we do is we look at our carbon 6. Our carbon 6 is facing upwards, so that means that it is D. Okay, now our anomeric carbon, the alcohol group, is facing upward. So they're both facing upward, and that means that they are cis, and this is beta. Perfect. So now, how many members are in this ring? Okay, there's 6 members in this ring. 1, 2, 3, 4, 5, 6. 6 members. 5 of these are carbon, and 1 is an oxygen. So that qualifies for the definition of a pyranose. Okay, a pyranose is a 6-membered ring, but it only has 5 carbons in the ring, and the other atom in the ring is an oxygen. Okay. Now B here. We look at carbon number 6. Okay, so this is carbon number 6. Okay, and it is going downwards. So we know automatically that this is an L. Now, this is our anomeric carbon, and the alcohol group that goes on it goes down. So they're both going down. That means that this is cis, and this is beta automatically. Okay, now let's look to see what definition of ring structure this fills. So there are 1, 2, 3, 4, 5, 6 members in the ring. 5 of them are carbons, and 1 of them is an oxygen. So again, it's a pyranose. It matches that definition. A pyranose is a 6-membered ring, but it only has 5 carbons in the ring, and the other atom in the ring is an oxygen. Okay, so here we have our carbon number 6 is going up, so that means that this is automatically D. Our anomeric carbon has the alcohol group going up, so that means that these are cis, and this is beta. Oh yeah, now let's see what definition this follows. So this is 1, 2, 3, 4, 5. This ring has 5 members. 4 of those are carbons, and 1 of those is an oxygen. So this matches the definition for a furanose. A furanose is a 5-membered ring, but it only has 4 carbon atoms in the ring and 1 oxygen. Okay, I hope this is getting easier. I know it was really kind of intense at first, but the more repetition you have, the easier it gets. Okay, so for this one, it's just basically definitions. Starch is made of 2 glucose polysaccharides, amylose and amylopectin. That's starch. Glycogen is a major storage form of carbohydrates in animals. Cellulose is not digestible in humans. Starch is a major storage form of glucose in plants. So glycogen and starch have similar functions, but glycogen serves a storage function in animals, and starch serves a storage function in plants. All right, nucleic acid structure. So remember your definitions of furanose. That's a 5-membered ring with only 4 carbon atoms in the ring and 1 oxygen. They're asking you to draw a beta furanose. DNA is deoxyribose, that's the sugar. RNA is ribose, okay? So I just included a picture from your book here to help you remember, like for nucleotides, you have your base, so it could be A, T, G, C, or U, and then you have your sugar, depending on whether you have deoxyribose or ribose, and then you have your phosphate group, okay? So here, this is how you draw it, and this is how you number your, this is deoxyribose, and I'll tell you why right now. But basically, you number 1, 2, 3, 4, 5, okay? And carbon number 2 is the most important one to determining which sugar this is. If there's an alcohol group here, then this is ribose. If there's no alcohol group here, then this is deoxyribose. Okay, and then they want you to be able to draw your ATGC. So here, I went ahead and included this picture from your book. Practice drawing these just a couple times. Know which ones are your purines. Pure as gold, okay? Pure as gold. That's how I remember the purines. And then the other ones are pyrimidines, okay? Notice that the pyrimidines only have a one-membered ring, and the purines have a two-membered ring, okay? So you need to be able to draw them, and you need to be able to know how they're connected to the sugar and the phosphates, okay? So for example, here you see guanosine 5' diphosphate. You have to have your sugar numbered because that's how you know which carbon to attach the phosphate groups to. And it's usually at the 5', okay? Okay, here, this is super easy, so cool. A bonds to T. These red lines right here, this is just one hydrogen bond, another hydrogen bond. So A and T have 2 hydrogen bonds. G and C have 3 hydrogen bonds, okay? So that just means that when you want to break them up, you need more energy to break these GC bonds because there's 3 hydrogen bonds as opposed to 2. So when you need more energy, that just means that you need a higher temperature, okay? So over here, they're asking you, oh yeah, so I gave you like an example sequence of DNA, okay? Look at all these G's and look at all these C's. That means you're going to need a pretty high temperature to melt this DNA strand, okay? Because remember, it's a double helix held together by those hydrogen bonds. And you want to break this up in order to start the process of transcription, okay? So here they're asking you, which of these lines, which of these curves goes with box number 2, which is this one. That's the GC. So if you have a high GC content, you're going to need a higher melting temperature. So this red line is the one that goes with that GC, box number 2. And this blue line, you need a lower melting temperature for that first box. On the x-axis, we have the temperature, and on the y-axis, we have denaturation percentage. And when they say denaturation, that just means the percent of this double helix that you were able to break apart. Okay, this is cool. So it's again super easy. They give you a DNA sequence, okay? 5', your DNA sequence, 3', and you're supposed to draw your messenger RNA. So all you do is you do the opposite. So instead of 5', you write 3', and then you write everything that matches this. But this is where students lose a lot of points. This is messenger RNA. RNA does not have T's. RNA has U's instead of T's. So for example, what matches this DNA T? A. What matches this G? C. C, G. G, C. A, normally you would say T. T matches the A. But because this is RNA, you would say U. Okay, so all of your T's that are supposed to be down here, you just turn them into U's. That's all you do. Now, the next question is, it's asking you what three-dimensional structure this forms. So this is where RNA can form some three-dimensional structures. So let's check it out. A binds with U, C binds with G, G binds with C, C binds with G, A binds with, I got lost, U, and then, okay, I got lost. But basically, you can draw the little arrows, and you can see, if you see this, this trend where these guys can bind with these guys, then that means that they will. And they form this little RNA hairpin structure. Okay, that's the three-dimensional structure that they're talking about. Okay, polymerase chain reaction. So here it's just PCR. They're just there's like 5 steps. So you just have to put them in order 1 through 5. So I went ahead and did that for you. And then here I got these pictures from your book so that you can practice drawing these sugars. So the most important ones are glucose, and then mannose, which is the epimer of glucose. It's the carbon 2 epimer of glucose, if you remember, because only that alcohol group on carbon number 2 is in a different position compared to the glucose molecule. And then here we have galactose, which is the C4 epimer of glucose. Yeah. Okay, we actually finished early. Does anybody have any questions? I'm going to go to the chat now. If you don't have any questions, you're welcome to leave. I'm going to go ahead and upload the key, my annotated key. I'll upload it right now as soon as class is over. And then hopefully by tomorrow, this video that I'm recording right now should be over. It'll be on CAD courses. You are welcome. Have a nice day. Okay, there's 8 people left. Does anybody have any questions? Okay, if there's no more questions, I'm going to go ahead and stop the recording here. Okay, have a good day, everyone.